In this video, we will continue our discussion about one more signaling pathways, is P13K and Akt signaling pathways, whether it will affect the EMT and cell survival in different matrices. So far, this paper had discussed two different pathways, the SMAD4 pathways and also the FAK pathways. Those does not affect that much on the EMT transitions in different matrices. So they checked that P13K and Akt also got changed when during this EMT transition. So they wanted to check whether this P13K Akt helps in this cell survival and EMT different matrices or not. For that, first they check the activated Akt in compliant and rigid cells at different conditions. And for control, they use the insulin, as insulin is known to trigger activated Akt, and they will use this as a background signal. And we can see that in the compliant matrices, the phosphor-Akt signals are low compared to the rigid matrices in almost in all conditions. And these things they have quantified in the left hand side of this panel A, where it is clear that in the case of this TGF-β treatment and in this compliant and rigid matrices, there is a significant difference of this activated Akt by total Akt ratio, or in simple terms, activated Akts are high in the TGF-β treatment in the rigid matrices containing cells compared to the compliant matrices containing cells. Thereafter, they wanted to see whether this pathways, P13K and Akt pathways, affects cell survivals on EMT or not. So for this P13K, they used commercial inhibitors, and for Akt, they used two different inhibitors. And what they are able to see is that in the case of first they validate whether the inhibitors is working or not, and we can see that in the case of this commercial inhibitors for P13K and also for this Akt, they are able to see less signals for this phosphor-Akt. And thereafter they checked whether these inhibitions of P13K and Akt affect apoptosis or not. So when they check the apoptosis, they are able to see that in the inhibitions of both P13K and Akt, they are able to see increase in the apoptosis in the rigid matrices, and it reaches to the level of this compliant matrices that we can see this panel C. And thereafter they also checked whether these inhibitions of the P13K and Akt affect the EMT transitions, and for that they checked the snail 1 as a marker or snail 1 mRNA expression levels, and they are able to see though these P13K inhibitions does not affect the snail 1 expressions, but the Akt inhibitions increases the snail 1 expressions, indicating that this Akt plays a role towards these EMT transitions in a way that it is negatively affect these EMT transitions. And similarly, they from this information they are able to find out that this Akt is suppressing these EMT transitions. So what it is, they overexpressed. Akt is suppressing EMT transitions, and also there is inhibitions of the P13K will leads to the increased apoptosis. So what they are able what they thought is they will overexpress this P13K to reduce the apoptosis, as by inhibiting P13K they are able to see increased apoptosis. So the idea is to overexpress P13K in the compliant matrices to reduce the apoptosis. So they use this P110 CAX, which contains the membrane region of this P13K, and they express using the adeno-associated virus, and they are able to see increase in the phosphor-Akt in the compliant matrices compared to the negative control, where they don't overexpressed this P110 CAX. And thereafter, when they check the apoptosis in the compliant matrices with this P110 CAX overexpressions, we are able to see reduction in the apoptosis when it reaches to the level of the rigid matrices, indicating that this overexpression of P13K helps in the reduction of this, apoptosis in the component matrices. However, the overexpression of P13K is not enough to affect the EMT changes, and that can be seen in this panel G. So for the EMT changes, they need to inhibit these AKT pathways. So overall, what they come to the point that is P13K and Akt affect the cell survival and EMT in the different matrices. And that is what they conclude from the results represented in this figure. And this is what their overall conclusions, that P13K and Akt pathways are affected in these different matrices in a way that will result in the cell survivals and EMT transitions differences in the different matrices. So that's all for this video, and also that's all for the final main figure of this paper. If you need to understand more about these experiments in detail and also wants to go through the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.